Go.
This is a whole muscle. A whole muscle is made up of a bundle of fascicles. A fascicle is a bundle of muscle cells or fibers, or a myofiber. The whole muscle is surrounded by epimysium, a layer of connective tissue. The muscle fiber is covered with a paramysium, another layer of connective tissue, and the myofibril is covered with an endomysium.
Action potential arrives at synaptic knob. Calcium channels in membrane open, allowing calcium to move into cell. Increased calcium causes synaptic vesicles filled with acetylcholine to fuse with the membrane. Acetylcholine is released from synaptic knob by exocytosis into the synaptic cleft. Acetylcholine is picked up by receptors on motor end plate of skeletal muscle. Potassium and sodium channels open, allowing sodium ions to enter muscle fiber. The increase in positive ions inside the muscle cell initiates an action potential on the sarcolemma. Action potential travels down sarcolemma into T tubules and stimulates the release of calcium from the terminal cisternae of the sarcoplasmic reticulum, where it has been sequestered by calsequestrin. Go.
The action potential drives down sarcolemma into the T tubules and stimulates the release of calcium from the terminal cisternae of the sarcoplasmic reticulum, where it has been sequestered by calsequestrin.
Calcium floods over the thin filaments and binds to troponin, pulling tropomyosin off the active site on the actin. High energy myosin heads leap to bind on the exposed active sites on the thin filaments, forming a cross bridge. The myosin head pivots to the low energy state, releasing ADP and moving the thin filaments over the thick filaments.
Mitochondria produces ATP energy.
The power stroke. ATP binds to the myosin head, causing the head to break free from the active site on the actin. The myosin head becomes energized as ATP is converted to ADP.
This is the thick filament. This is the thin filament. This is the Z disc. This is the inline. This is the I band. This is the A band. And this is the H zone.
Calcium is pumped back into the sarcoplasmic reticulum and attaches to calsequestrin, allowing tropomyosin to once again cover the active site, resulting in relaxation of the muscle.
And the acetylcholinesterase flows in and breaks up the acetylcholine into acetin and choline, which then flows back into the axon and goes into the synaptic vesicles and makes acetylcholine.